We briefly introduced the lab bench in a different video, one that used Nobel Prize winning CRISPR experiments to demonstrate principles of restriction enzyme digestion and DNA electrophoresis. This tutorial uses a simpler example with lambda DNA digested by 3 restriction enzymes, and its purpose is to demonstrate additional features of the lab bench. To get to the lab bench, you click the button at the bottom of the button bar from the data screen. So the first thing we need is to get some DNA samples. Go to the DNA menu, click that, say Select DNA Samples. And the ones that we need are in a folder called Wet Lab Simulations. And we'll use the Lambda DNA folder and select Lambda DNA. On the lab bench, there's a feature where if you need more than one identical sample, all you have to do is click on this line in the Open in Process window. Go to the Site menu, and you can open up to 4 enzyme tubes, or you can select enzymes from a list. And just as a quick aside here, I'm going to temporarily go back to the data screen by clicking the button at the bottom. You can do the same thing on the data screen, but one additional feature is this colored button allows you to select enzymes, and it shows you whether it's a straight cut or a stagger cut and the actual nucleotides that are part of that site. Going back to the lab bench, go to the Site menu, and we'll go ahead and open up the ones that are already in the CASED folder. In the Lambda DNA folder, it's inside the CASED folder. So what we will do is shift-click and open these 3 enzymes. Next, we need to add the enzymes to those DNA tubes. If you go to the Tools menu, you can open all the tube caps. You can also open and close them by double-clicking on the caps. So I have a fresh tip on this pipette, and notice when the tip goes into the tube, you see in this message, enzyme A contains enzyme BAMH1. So I'm going to click the plunger, and I'm going to add that to DNA tube number 1, and I'll click the plunger to release it. After a tip has been used, it should be ejected from the micropipetter. For the purpose of the simulation, all you have to do is touch the biohazard box to remove the tip. In reality, I'll just go to the Tools menu and show you the side view of the micropipetter. In reality, you would use this second ejection button to eject the tip. So now we're going to add a second enzyme to that first DNA tube. We'll get a clean tip, and we'll go to this particular enzyme. Now notice when it makes contact, it says enzyme B contains the enzyme EcoR1. What we need to do is click the plunger. Now watch the plunger carefully and notice it only goes down part way. That's to the first stop. So in reality, that's what you would do with a micropipetter. You'd go down to the first stop and then release, which would suck liquid into the tip. And then we will go to DNA tube number 1. And now watch the plunger carefully, and it goes down all the way. So you'd go past the first stop all the way to the bottom to eject that liquid into the test tube. So we've used that tip, so it needs to be ejected. Again, you would in reality, you would push that ejector button. Get a clean tip. Going to go to enzyme C, which is enzyme Hindi3. Go down part way, suck that up, depress it all the way to put that enzyme into the DNA tube. So now this particular DNA tube, if you just move your cursor over it, you will see a message appearing up here. DNA1 contains lambda DNA and the enzymes BAMH1, EcoR1, and Hindi3. Get another clean tip, and this time we will add the enzymes BAMH1, and EcoR1. To DNA tube number 2. And to verify that, move your cursor over the tube and you can see up at the top the DNA2 contains lambda DNA and the enzymes BAMH1 and EcoR1. Get another clean tip and just add enzyme Hindi3 to tube 3, DNA tube 3. We'll add a fourth DNA tube to serve as a control, and an easy way to do that on the simulation is to simply click one of these lines, the existing DNA lambda lines. So a new line appears in the open and process window, and that DNA tube number 4 then appears in this location on the lab bench. The next step is to put a heat block on the lab bench. So I'll go to the Tools menu, Heat Block, and show it. What we need to do is to add these tubes to the heat block. And to do that, it's quite simple. All we have to do is drag them one at a time to the block. Now we need to close the caps. That would actually happen automatically when you dragged it to the heat block. But just for completeness, we'll say close all tube caps. And I'm going to put these 4 DNA samples in the heat block just by dragging them. Click for top view of the power switch on the heat block, and there are the 4 DNA samples. Turn on the power switch, and in reality this would be heated for a specified period of time, but in the simulation it's going to happen instantly. Click to remove the tubes, and the enzyme tubes are positioned up here to get them out of the way, and here are the 4 DNA tubes that have been removed. Go to the Tools menu, and for simplicity we're going to use the Quick Setup, which brings the gel box and the power source onto the lab bench. We need to load the gel, and we'll start with DNA number 1. We again could double click, but it's easier to just open all of the tube caps, get a fresh tip, and go to DNA tube number 1 very carefully. Click the plunger, and move the tip down until the yellow message appears. Click the plunger, and you will see that the sample appears here. In reality, loading dye would have been added. That was not shown in this simulation. So we need to remove that tip, and you want to very carefully just touch the box without touching the DNA tubes. Get a fresh tip, go to DNA tube number 2, and transfer that to well number 2. Go to tube number 3 and put that in well number 3. It can be a little tricky to do it. And then go to tube number 4 and add that to well 4. Double click on the gel box to see a side view of it. Double click again to see the end view. Now we need to run the gel. First, we need to attach the lid and the wires that conduct electrical current from the power source. So now it's all set up to run. And if you click this toggle switch on the power source, the fragments will move across the gel. I'm going to call those ghost fragments. In reality, you wouldn't see them. In reality, I'm going to click this clear button, this white button, and in reality, you wouldn't see them at all. I'm going to put them back and click this purple button to stain the gel. In reality, you couldn't do this, but on the simulation, you can reload these fragments, leave them stained just so we can see how they move across the gel. They're moving in a very realistic fashion. There's formulas that cause them to move just the way they would on a real gel. This was an hour run. Notice it's 60 minutes. It says in 100 volts on the power source and 1% agarose. All of those things can be changed. If you wanted to change the time, let's say 30 minutes, double click on this field, type in 30, click the set button, set time for gel blot run. Then you can reload the gel and run it for 30 minutes. Or you can go to the run button and do an hour run to get us back to where we were before. You can also change the voltage setting on the power source by clicking this round button. So it says 100, that's the default 100 volts. I'll click that. I'll make it 75 down to 50. Reload the gel. And you'll notice they don't go nearly as far because there's not as much of a difference to cause those fragments to be attracted to one end of the gel. Click that again, put it up to 125. As you repeatedly click that button, it toggles between the various options. So it says 125, reload it, and now it's going to go much further because there's more of a voltage difference between the two ends of the gel, more of a difference in charge. Go back to the hour run, and actually I'm going to click that, go back to 100 and run that again, just to get back to where we were before. 1% agarose, 60 minutes and 100 volts. And if you go to the run menu, rerun with an agarose concentration of, let's say, 2%. And the gel fragments don't go as far because they have to fight through that denser agarose concentration. And I'll run it again at a default setting, and this time I'll just say run, timed run. So we're back to where we began. Distances for the various lanes are shown here. This is how far they've gone from a scale of 0 to 100. On the gel menu, you can determine what the sizes of those bands are. So for example, for lane 1, these would be the sizes, the kilobase sizes for these various fragments. And then you could use the gel menu to do it for the other 3 lanes. There are so many more things that you could do on the data screen. Click the data screen button, and notice this white gel blot button indicates the gel is hidden. I'll click that to show the gel. I will do an hour run. Click these lanes to show the sizes in kilobases of the various fragments. Or you could set it back to distance moved, either way. You can also click on individual fragments on the data screen. they turn red, and then that highlights that particular fragment, and it says 564 base pairs. Click the sequence button to see those actual base pairs of DNA. Here's another one, 9,416 base pairs. Click that sequence, and there's the sequence. You can magnify the gel to make things easier to see by clicking this yellow magnifying icon, and do the same thing. Some of those are not visible on your screen right now. There, for example, the first ones. In some cases, it will be difficult to see individual fragments because of the overlap. So there is a button here to make them narrower. When you click them, they go back to that original default display. You can also click in a lane, anywhere in that lane. And after you've done that, if you hold your cursor over a fragment, it will tell you its size and base pairs. But you do have to click in the lane first, like the first lane. That one is 493 base pairs. Click this one, this fourth lane. That's 6,557 base pairs. That procedure also works if you click on this button to make the fragments narrower. For example, 2284 for that particular fragment. And if you click on another lane, you're going to have to go back and make it narrower again. Click the magnifying glass icon again to go back to the original view of the gel. And just like on the lab bench, you can change the color. Photo button is grayed out. That feature no longer works, unfortunately, and it gives a message about using screen capture. That message may not be totally up to date with modern screen capture, but there are a variety of methods on both the Mac and the Windows operating systems for capturing the screen. You can just Google those if you're not familiar with them. Next, we're going to repeat that same analysis with Lambda DNA, but this time use the data screen instead of the lab bench. The data screen is much faster, and it also has many more capabilities than the lab bench. I cleared the screen using the clear menu. So to repeat that analysis, we will go to the DNA button of the silver button bar and open the Lambda DNA. Next, go to the enzyme button and open those same three enzymes that we used before. Shift-click to highlight the three enzymes. Go to the Cut DNA button, and this time we're going to select with multiple enzymes in the same digestion. What that does is it takes basically three different Lambda DNA samples, and it digests the first one with the first enzyme, which is BAM-H1, and then there's a double digestion of a different Lambda DNA with BAM-H1 and EcoR1, and then finally a triple digestion of Lambda DNA with BAM-H1, EcoR1, and Hindi3. If you recall, the first DNA tube we had on the lab bench contained these three enzymes. So instead of using the Quick Load Run button, I'm going to click the Load button on the data screen. And when that button turns blue, that indicates it's been loaded. The second DNA tube that we had on the lab bench had BAM-H1 and EcoR1. So I'm going to activate that by clicking that line in the Open in Process window. Click the white button next to the number 2. That represents well 2. And we've selected it. When you select it, it's purple. When you click load, it turns blue, indicating it's loaded. The one thing we're missing here is Lambda DNA digested by just Hindi3. So I'm going to make this window a little bit bigger. I'm going to activate Lambda DNA and then activate Hindi3. Go back to the silver button bar and this time say with a single enzyme. And then I will click the white button next to the number 3 and click the load button. So then well 3 has been loaded. I just made a common mistake that I want to show you how to correct. I had neglected to click this line, lambda DNA digested with Hindi 3, before I clicked the load button. And that's the reason why the enzyme is not showing here. The gel would actually run, it actually is loaded with the digested lambda, but it just won't show the proper label in the loaded window. So to fix that, I am going to select well 3, click the clear button to clear it, and notice that line disappeared from the loaded window. And this time I'm going to click on DNA with the digested by Hindi 3 and reload it, and now it's showing up properly. Finally, we had a control on the lab bench, so I will take the undigested lambda DNA and put that in well 4, click load, and this time we'll use the run button on the data screen and say run DNA or protein gel. Stain it, and if you compare that with the result we had on the lab bench, it's identical. One thing I did not talk about earlier was how to label the gel. You could use this label under the gel. There are some features that help you do that, but it's small and it's cumbersome to use. I have found it to be much easier to simply use the loaded window as a label. So to demonstrate that, I'm going to move everything over just to give me some more room, and take the loaded window, bring it into position, and make it large enough horizontally so that you can get everything on a single line. And then you can see that the triple digestion is in well 1, the double digestion is in well 2, the single digestion is in well 3, and then the control is in well 4. Once you've done that, the easiest way to save your results is to use the screen capture capabilities of your computer to capture whatever part of that image you would like to save. Notice also the number of fragments in each lane. There are 20 in lane 1 that you can see right here. And you can also see those individual fragments down here. There are, if you click on lane 2, there's 14 fragments in lane 2 corresponding to these numbers here. Lane 3 has 8 fragments and 8 down here, and then 2 fragments in lane 4. Now notice, going back to the first lane, for those 20 fragments, you can scroll across and see the sizes of those, or you can click on individual ones. You can click the sequence box and then scroll through the various sequences of each one of those individual fragments. And finally, let's say that you were interested in determining if this really was lambda DNA that we were working with. Well, one way to do that would be to click on one of these fragments in the control, show the sequence, and then highlight part of that sequence. Then right-click with your mouse and go to the copy, selected text, to the clipboard and open the NCBI blast site. So this will automatically open that web page. And then you can click in the field, right-click, paste the sequence, scroll down, click the blast button. So here are the blast results, and if you scroll down, you see there are very low E-values, very high percent identity. And if you scroll down, you see lambda virus complete genome. See the separate video tutorial on the integration of KCIT with Mega Bioinformatics software. and also visit the CASEIT website to see additional tutorials on sequence analysis and other bioinformatics capabilities.